Okay, so I'm in the interest of time, so each speaker has 5 minutes. We're going to have everybody do their presentations at the beginning. We've got a couple people trying to have a tight schedule, and then we'll take questions at the end. So our first speaker is Tony Choueiri, who's going to be presenting a tetravalent PD-1 VEGF bispecific antibody for treatment of renal cell carcinoma.
Thank you, Naomi. Thank you all for staying day 2 here. I don't know where, probably look down. So let me tell you about this drug now from Pfizer. That's why you have the PF, long names. I'm gonna call it 404, just to, or the bispecific, for renal cell cancer that's been explored in a study that is open. Let me start by telling everyone, despite I'm a big believer, is that you add a drug in theory to a doublet or a fourth drug to a triplet, I think you believe you achieve better outcomes, and that's why we have cured many diseases, from TB to AIDS to many other, to leukemias, to lymphoma. But in renal cell, 3 versus 2 did not pan out for many reasons. Cosmic 313 was a positive study adding cabozantinib to nivo-ipi, but the overall survival was negative, and the side effects profile from the triplet was higher, and LIGHTSPARK-02, to the surprise of many of us, was a negative study. We could not add benefit from BEL or the CTLA-4 inhibitor Q to the combination of, to a powerful combination of pembrolizumab, and you're gonna see the data later this year. So what can we do? So let me tell you a bit about 404, which is a tetravalent antibody. The whole premise about this, what I call topography of bringing PD-1 and VEGF close together, is the simultaneous, and this is again very theoretical and also preclinical, binding of PD-1 and VEGF would amplify therapeutic potential, especially of the PD-1, PD-L1 binding. I'm going to show you data that cooperative binding will improve PD-1 inhibition in the presence of VEGF, because part of the antibody gonna bring the VEGF closely. And VEGF by itself, inhibiting VEGF, is a therapeutic target. Here the antibody is functionally enhanced arm with, in theory, normalized tumor vasculature and also reduce VEGF-related immunosuppression. And that antibody used the well-known IgG4 backbone that is consistent with approved anti-PD-1 therapy. So that is a preclinical experiment that shows that soluble VEGF increases this functional blockade of PD-1 through cooperative binding. You can see here, and I'm going to use arrow here, without VEGF in the milieu, with VEGF, you can see how the combination of the 404 here with VEGF lead to significant induction of that PD-1 inhibition. On the other hand, you have, if you want, a control arm with pembro only and pembro plus VEGF as a control arm. I wish this was a clinical trial, etc., that is less. So will this lead to an enhanced clinical benefit, and in which watch patient? I think this is a very reasonable, very definitely a reasonable question. Now, also this data shows that preclinically 404 has the potential to be best in class also VEGF inhibitor. This is a HUVEC system we use a lot. We can discuss if this is the right one or not. But this is, for example, bevacizumab here in black, in red Ivo. That's the compound from Summit that is PD-1 VEGF, the first one, the most evolved, where the phase 3 are reading. And that's 404 compound here. This is the percentage of growth inhibition. And you can say that depending on the concentration, that is really different. And you can achieve a growth inhibition, let's say at 50%, way less with the bispecific here. And it's around much higher versus, let's say, in this assay, with the limitation of this assay with Ivo or bevacizumab that you wouldn't expect without the PD-1. It's definitely at least more potent. And based on that, this is the symbiotic G U08 study that is open in many centers here, where we have a cohort of untreated patient first, but monotherapy, a cohort in combination with IPI, and another cohort in combination with axitinib. And of course, a design like this will let us add multiple cohorts. So now the phase 1, many of the trainees learned 17, 18 patients, could be 300. The key eligibility criteria, this is be used in untreated clear cell RCC. Nothing unusual about the endpoints. We're going to look at toxicity, tolerability, but also, of course, the secondary endpoint of response rate, durability of response are going to determine how we move with what trial, with what phase 3. So again, hopefully this overcome the triplet challenge. I didn't say it's less toxic or what, the hope. This drug has a unique profile from preclinical, and now it's being in development. I hope your center is a site for this study. Thank you.
All right, good morning, everyone. I first want to thank the organizing committee for inviting me to speak on behalf of my co-authors and to present our study titled A Phase 2 Study of Total Consolidative Metastasis and Primary Directed Therapy for Renal Cell Carcinoma. As many of you are aware, frontline immunotherapy-based treatments have transformed outcomes for patients with metastatic RCC. However, complete responses remain rare, with only 9 to 16% of patients able to achieve a CR. And so for the majority of patients with stable disease or partial response, they often progress, and so there's a need for better strategies among this patient population. We hypothesize that a consolidative approach after 6 to 12 months of immunotherapy-based treatments may be effective. And that's because, one, when progression does occur, usually occurs in pre-existing lesions, and two, the deepest response to IO-based therapies typically occurs at a median of 7 months. This is a single-arm phase 2 study being conducted at a single academic center, and we are enrolling patients with metastatic clear cell RCC who have had 6 to 12 months of time elapsed since they started their IO-based therapy. Now, this IO-based therapy could either be IO-IO therapy or IO-VEGF-TKI therapy. We have 2 pre-specified cohorts and hope to enroll 8 patients from each cohort. At the time of enrollment, at that 6 to 12-month mark, patients must have achieved stable disease or partial response and must have no more than 5 metastatic sites. After enrollment, patients will discontinue their systemic therapy and then undergo total metastasis and primary directed therapy, which involves any combination of radiation, surgery, and/or ablation to eradicate all residual disease. Afterwards, patients will not immediately resume systemic therapy, but rather undergo observation and restart systemic therapy at the discretion of their treating oncologist. We are primarily aiming to estimate the proportion of patients who are able to achieve at least 12 months of treatment-free survival, and we define treatment-free survival as the duration of time from stopping systemic therapy until restarting systemic therapy or death. And we chose treatment-free survival as our primary endpoint because we believe it represents an endpoint that is meaningful to patients, given that it's capturing the amount of time that patients have adequate disease control without needing to go back on systemic therapy. Secondary endpoints include recurrence-free survival and the rate of grade 3 or higher adverse events. We are powering our study against a historical benchmark of 13% of patients achieving at least 12 months of TFS after achieving stable disease or partial response to frontline IO-based therapies, and we aim to measure an improvement to 34%. And 24 participants gives us 80% power using a 1-sample log rank test against that historical benchmark. The study also involves exploratory correlative component, where we are collecting blood and tissue samples both before and after patients undergo this consolidative approach. And in particular, we're interested in correlating immune signatures and nuclear speckle states with improved treatment-free survival. One immune signature that we're particularly interested in are progenitor-exhausted T cells, which is a self-renewing pool of T cells that has been shown to be reinvigorated by anti-PD-1 therapy and have been linked to longer duration of response to immunotherapy and melanoma. And meanwhile, nuclear speckle states are gene-activating substructures that can exist in normal or aberrant states, and aberrant nuclear speckle states have been shown to correlate with worse outcomes in RCC. Currently, there's no prospective evidence that demonstrates that this type of consolidative approach can benefit patients after achieving stable disease or partial response to frontline immunotherapy-based treatment. So we hope to fill that gap in the literature. And we're doing so using what we believe is a patient-centered endpoint in treatment-free survival. Since it's a single-arm, single-institution study, it may provide foundation for a larger multi-site RCT. And ultimately, we aim to re-envision residual disease as something that is not simply treated indefinitely with systemic therapy, but rather may potentially be eradicated in the process by patients' meaningful time off of systemic therapy. Thank you.
Okay, our next speaker is Abrahami Das, and she's going to phase 2 study of cemiplimab and FIAN, the LAG-3 inhibitor, following SBRT for oligometastatic clear cell.
Hi everyone, good morning. I am a PGY2 internal medicine resident, so bear with me, but I'm going to be talking about a phase 2 study evaluating cemiplimab with and without fianlimab following SBRT for oligometastatic renal cell carcinoma. So oligometastatic renal cell carcinoma is a distinct subset within the umbrella of metastatic RCC. It is unique because it's an intermediate stage between localized disease and widespread metastasis. This means that we have room to employ localized therapies such as SBRT, metastasectomy, and cytoreductive nephrectomy in certain patients in combination with immune checkpoint inhibitors or other systemic therapies. So in our study, we're evaluating SBRT followed by a dual immune checkpoint blockade. So why SBRT? The largest study that we have to date that kind of evaluated SBRT without any systemic therapy in oligometastatic RCC showed a 1-year PFS of 67.5%. Providing SBRT before immune checkpoint inhibition can improve its efficacy because of immune cell activation, proliferation of CD8 plus T cells, as well as causing tumor cell apoptosis and upregulation of PD-1 receptors. Immune checkpoint inhibition following SBRT and following metastasectomy has already been studied and evaluated. We're trying to build on this literature and look at SBRT followed by dual immune checkpoint blockade. So the dual immune checkpoint inhibitors that we're using here are PD-1 inhibitors and LAG-3 inhibitors. PD-1 and LAG-3 are the most commonly upregulated receptors on tumor-infiltrating lymphocytes in clear cell RCC. They act through 2 distinct mechanisms, drive T cell exhaustion, and some studies have also found that blocking PD-1 can cause overexpression of LAG-3 as well. So it kind of tells us that PD-1 inhibition alone may not be enough to restore anti-tumor immunity. In our own multi-omic analysis, we found that high LAG-3 expression, it can be linked to and is associated to increased median overall survival compared to lower LAG-3 expression in RCC. It's also associated with higher tumor mutation burden as well as more immune infiltration. All this kind of points towards a hotter tumor microenvironment and kind of tells us that LAG-3, through certain immune evasive mechanisms, could be conferring resistance to PD-1 receptors. For our study design, we're going to be looking at oligometastatic renal cell carcinoma. It has to be clear cell. Variant histology will be excluded. They will undergo SBRT to both primary and metastatic sites, undergo randomization, following which they will be randomized to Arm A and Arm B, Arm A with cemiplimab and fianlimab, and Arm B with cemiplimab alone. Our primary endpoint here is going to be 1-year progression-free survival, and our secondary endpoints are mentioned on this slide. So for our study design, furthermore, based on our calculations and with a hazard ratio of 0.6, we have calculated 32 evaluable patients in each arm, coming up to a total of 64. We anticipate a 10% drop-off rate, so coming to a total of 70 patients with 35 in each arm. They will undergo something stratified randomization, and this will be based on IMDC risk score, CNS mets, whether they're present or absent, as well as the number of metastatic lesions, whether 1 to 3 or 4 to 5. Sites include Stevenson Cancer Center, University of Nebraska, Ohio State, as well as Sarah Cannon Research Institute through Rocky Mountain Center. So in conclusion, to our best knowledge, LAC-BOOST trial is the first phase 2 trial evaluating SBRT followed by a LAC-3 inhibitor and PD-1 inhibitor. It is a unique state with less aggressive tumor biology and more robust immune surveillance, and we're trying to capitalize on this intermediate stage and improve immune checkpoint inhibitor efficacy in this stage. Thank you so much.
All right, next up we have Dr. Rana McCabe from UC San Diego discussing the PRISM study.
Wonderful, thank you all so much. This is our phase 1b IIT of retifanlimab with ruxolitinib in solid tumor malignancies progressing on a prior checkpoint. Here are my disclosures. And just as a way of introduction, we know that ICI resistance is common in RCC, seen by the majority of patients, and options after ICI progression really remain an unmet need. MDSCs, as we know, suppress the tumor anti-immune response, and actually ruxolitinib, which has been utilized across solid tumor malignancies, can actually reprogram them and restore T-cell function. This is work that's been led by the Zach Lab, which actually has recently transitioned to the University of Utah, and work published in Science in 2024. Ruxolitinib with anti-PD-1 actually was able to achieve a 53% response rate in ICI refractory Hodgkin's lymphoma, obviously a different setting. And preclinical models have actually demonstrated in renal, in RCC, in colorectal cancer, in lung cancer, that actually adding ruxolitinib with checkpoint inhibition can elicit a tumor response. So our hypothesis was actually adding ruxolitinib with a PD-1 can overcome acquired ICI resistance and result in an improved objective response rate on treatment. So here is the schema for our study. It is enrolling patients with non-small cell lung cancer in addition to clear cell RCC having measurable disease. Patients need to have been exposed to one and only one prior immune checkpoint inhibitor and need to not have primary resistance to that prior checkpoint inhibitor. They need to have had either stable disease or a response and then subsequently ultimately progressed. Patients will be enrolled. We're currently in dose escalation. We've designed this study with both a dose escalation, de-escalation component so we can be efficient in our phase 1. Starting patients at the 15 milligram dose of Rux paired with retifanlimab, which is a PD-1 inhibitor. Retifanlimab dosing will be fixed, and we will up-titrate Rux to 20 milligrams, and if we run into any issues, have the opportunity to de-escalate, though we don't anticipate transitioning across all of the dosing cohorts. Once the dose is identified, we will conduct an expansion to assess response, which is a key primary endpoint with the study powered for response, with also stratification based off of type of malignancy, setting the null at 10 and alternate hypothesis at 30%. This study is also enriched for tremendous biomarker correlatives that have been actually supported by Insight, which is funding this trial. They've been actually a great partner with baseline biopsy that happens ahead of a ruxolitinib lead-in after 7 days, and then patients initiate combination therapy. There'll be serial bloods. There'll be an on-treatment biopsy that'll be collected and processed at the University of Utah Zach Lab. So here is our vision and impact. So, this is really an unmet need. I think provides the hope is that this study will provide an early signal of adding JAK inhibition, which is a novel mechanism in the RCC space with anti-PD-1, and potentially reprogramming the immune response is certainly an unmet need. I think the other beauty about this trial is just mechanistic insights with the baseline and on-treatment biopsies and paired tissue samples with robust correlative science. And I think the mechanism in the lab, preclinical studies, has been recapitulated across several tumor types preclinically, and I think there's the opportunity to expand. We actually had to really narrow just to RCC and non-clear cell, though there was initial discussions around just having it be a pan-solid tumor. But for us to really be able to assess a signal in RCC and a strategy for moving forward, really wanted to hone that in. So with regards to informing next steps, we hope that this study will inform the safety and tolerability and identify the early response data to help inform potentially a randomized study with JAK inhibition in this context. Thank you.
Okay, I don't think my mic is working, but the next one is Sahil Doshi, who will be speaking from Memorial Sloan Kettering.
Thank you to the KCRS committee for the opportunity to share our trial in progress today. I'll be presenting on a phase 1b/2 study of abemaciclib plus cabozantinib in immune checkpoint blockade pretreated metastatic clear cell renal cell carcinoma and translocation associated renal cell carcinoma. So there is a need for novel systemic therapies in patients with metastatic renal cell carcinoma who progress on standard agents. Our study is focused on 2 distinct RCC subtypes with a biologic rationale for inhibition with a CDK4/6 inhibitor, such as abemaciclib. First, for clear cell RCC, a hallmark of clear cell RCC is von Hippel-Lindau inactivation, leading to HIF-2 alpha accumulation. This affects multiple downstream targets, including VEGF, and also leads to increased cellular proliferation via activation of the CCND1 gene, which encodes cyclin D1, a key partner to cyclin-dependent kinases 4 and 6, in driving cancer cells through the cell cycle. In previous RNA-based analyses for patients who underwent nephrectomy, we found that CCND1 levels were independently associated with an increased risk of recurrence. Further, we conducted a preclinical study investigating the combination of cabozantinib and abemaciclib in a clear cell RCC patient-derived xenograft mouse model, and found significantly improved survival with the combination of the 2 drugs, as shown on the curve to the right. Second, for translocation-associated renal cell carcinoma, these are rare variants that lack consistently effective treatment. A recent report from Dr. Vishwanathan's lab at Dana-Farber demonstrated that downstream effects of the translocation RCC gene fusion transcripts include cyclin-dependent kinases. In fact, in those studies, they showed that preclinical models of human translocation RCC demonstrated CDK dependency and anti-tumor effect with CDK4/6 inhibition. For those reasons, we hypothesized that targeting these pathways in these 2 RCC subtypes will provide clinically meaningful benefit in treatment refractory disease. So this is an investigator-initiated, single-arm phase 1b/2 2-center study that is being conducted at Memorial Sloan Kettering Cancer Center and Johns Hopkins University. We're evaluating the combination of multi-receptor TKI cabozantinib with the CDK4/6 inhibitor abemaciclib. Patients must have received both an immune checkpoint blockade and TKI therapy previously to be eligible. In the phase 1b dose escalation portion of the study, we will enroll between 4 and 24 patients in a standard 3+3 dose escalation design across 4 separate dose levels. Patients will be treated with once daily cabozantinib and twice daily abemaciclib. Imaging will be performed every 8 weeks for the first 3 assessments and then every 12 weeks thereafter. We are also looking at pharmacokinetic assessments for both drugs during phase 1b. The safety information from phase 1b, as well as the PK results and early efficacy signal, will be used to inform the recommended phase 2 dose. In phase 2, we will apply this dose using a Simon 2-stage minimax design, with the primary endpoint being objective response rate at 24 weeks. In stage 1, if 4 or more of the first 12 patients have an objective response, we will expand the study to a total of 25 patients. We are currently enrolling patients in phase 1b at dose level 2. This study addresses a critical unmet need by rationally targeting convergent oncogenic dependencies, VHL-driven VEGF signaling, and CDK4/6 mediated cell cycle dysregulation in 2 distinct RCC subtypes. We believe the inclusion of translocation-associated RCC is especially meaningful given its rarity, often poor prognosis, and near total absence of molecularly driven treatments. This trial is among the first to clinically exploit the CDK dependency recently identified in translocation preclinical models. Further, we'll be looking at archival tissue for these patients and through molecular analyses looking for markers or signs of CDK response to these inhibitors. If successful, this combination could lay the groundwork for broader exploration of CDK4/6 inhibition across one or both of these RCC subtypes. Thank you to all the patients who are participating in this study currently, my mentor Martin Voss, Yasser Ged, the site lead at Johns Hopkins, and all our co-authors.
All right, last but not least, we have Dr. Pavlos Masoul from MD Anderson, who will discuss the STAR RMC's trial.
Thank you for the opportunity to discuss the STAR-MIC trial in patients with SMARCB1 deficient renal medullary carcinoma that have been previously treated with one prior therapy. As many of you know, RMC is extremely aggressive, used to be the deadliest kidney cancer in adults. Now we've got better outcomes thanks to therapies that we've developed, but it still remains a deadly disease. It's the only cancer that I'm aware of that so specifically afflicts a minority, young individuals of African descent, because of its connection with sickle cell trait and other sickle hemoglobinopathies. And it's always defined by the loss of the SMARCB1 tumor suppressor. The anti-VEGF therapies developed for clear cell renal cell carcinoma do not work at all against standard RMC. And the standard immune checkpoint therapies that work against many other cancers do not work against RMC and may actually feed the disease, making it grow faster. And the cytotoxic chemotherapies that do not work, for example, against clear cell renal cell carcinoma can occasionally work against renal medullary carcinoma, but in the first-line setting produces responses of less than 30% with progression-free survival of less than 4 months in the first-line setting. And what our comprehensive molecular profiling has identified, along with our preclinical studies, that TROP2 is a cell surface target in renal medullary carcinoma, and that topoisomerase 1 inhibition can be a viable therapeutic target in RMC, in our RMC preclinical models. And subsequently, we repurposed sacituzumab govitecan, the FDA-approved agent in, for example, breast cancer targeting TROP2, in heavily pretreated patients, 4 of them. These are patients that had been treated with a median of 3 to 4 prior therapies, including the panitumumab-based therapies that we presented as an oral presentation at this year's ASCO that was transformative for the patients with RMC. So these patients had progressed to the panitumumab-based regimens and received sacituzumab govitecan. One had progressive disease as primary response, but there was 1 partial response and 2 stable diseases with a median PFS of 2.9 months. For RMC, especially for such heavily pretreated RMC, this is intriguing. Hence why we designed and activated the first patient who will be treated right now, today, in the next 2 hours, in the STAR-MIC trial, just signed the order, that uses sacituzumab tirumotecan for patients with RMC. The antibody is the sacituzumab that is used in govitecan as well, but this is the next generation because it uses a different linker that is more stable and potentially can increase the release of the payload more to the tumor cells and the tumor microenvironment, increasing potency and having a better therapeutic index. The payload, as opposed to the irinotecan derivative SN38 that govitecan uses, is a belotecan derivative, but again, a topoisomerase inhibitor. Eligible patients will be patients with previously treated RMC with at least 1 prior line of therapy. This is the trial design. The primary endpoints are the ones that we have used successfully in all of our prior RMC-specific investigator-initiated clinical trials like this one. The design parameters are the ones that we have been using again successfully for the prior clinical trials and can give us quick inferences. These are the same type of designs that I presented at GU ASCO. These are the ones cited as well in the January this year draft guidance by the FDA on novel trial designs. What I discussed on that talk, Boeing design, BOP, TOP2, this is the time to event Bayesian optimal design. That's the one that this trial is using, again, to increase the efficiency and the rapidity of our inferences in this state. And these are, again, the parameters where we have the 2 co-primary endpoints of objective response rate and disease control rate. And always, we are ready to learn. No matter what ends up happening clinically, we will learn from this trial by anchoring correlatives, tissues, and blood samples that will let us understand why some patients might respond or why they might not. And this will be connected to the samples from all of our prior RMC-specific clinical trials with different mechanism of actions to help us learn not only about RMC, but about sacituzumab tirumotecan as well. And the vision is, again, to accelerate renal medullary carcinoma research. As many of you know and have seen versions of this vision, when it was first described in 1995, the median survival of RMC was 4 months from diagnosis. For these young patients, this is devastating. They're otherwise healthy. By 2017, when the first ever funding for RMC was given, it was the ASCO Young Investigator Award supported by the Kidney Cancer Association, the median survival was 13 months from diagnosis. And we said in the AACR kidney cancer meeting in 2023, openly so that we are accountable to our patients and our stakeholders and our supporters, that we're going to get this to 2 years by the end of 2025. As you guys know, and these are published data, we have been able to achieve this acceleration of these outcomes. At the same time, we said that we're going to get the median survival to 5 years by the end of 2030. Again, in that presentation, how? Through these investigator-initiated clinical trials. 3 have been completed already. 1 is ongoing, shown here. And the 5th one now is this STAR-MIC trial that will give us the information to hope to get there, the biological insights and rigorous data science principles guiding the therapy development to get to our end goal, which is to end renal medullary carcinoma. Thank you for your attention.
Okay, so we have about 8 minutes to answer questions. Monty?
Thanks for your presentations. Sahil, I was really interested in your study. I think it was maybe 2 years ago here that Brad had presented some of the data for Abema and clear cell RCC without, a very compelling signal. Any thoughts to maybe making this more of a TRCC focused study as opposed to something that encompasses clear cell as well? Just a little skeptical if we didn't see any monotherapy activity to see synergy.
Thank you for that question. So, I believe that was presented 2 years ago here, and it was focused on clear cell RCC, a relatively more kind of heavily pretreated population. I think a few thoughts in response to that. The first, I do think so far the data with CDK4/6 inhibitors and clear cell RCC have been somewhat disappointing, but here we are looking at it in combination with the TKI, which has not been done so far, and we're bringing it a little bit earlier into the clinical setting where patients can have had, even just 1 line of therapy if they've had both immune checkpoint blockade and TKI in that setting. The second, we are also, with the recent data that was presented last year here, we included the translocation RCC into this study, and so we're hoping that this does have a different biologic rationale for including the CDK4/6 inhibitors. We might see early efficacy there, and we could expand further into that subgroup. And then finally, I think one point that we've been thinking about, with abemaciclib monotherapy, there was no objective response rates seen on that small study, but if you take, for example, everolimus, which has had some response, but as monotherapy is not very clinically efficacious, but then when you combine it with lenvatinib, it's very efficacious. So there is a possibility that we see that sort of synergy as well here.
My question is for Reina. Hey, so the ruxolitinib data. Folks who have had polycythemia probably have been put on this drug for some time, or an alternative JAK inhibitor. Is there any clinical evidence that they tend to have better responses to checkpoint blockade if any of them have cancer? Like Chuash patients with VHL disease?
I don't know that I have insight into that data. It's a good question, and you're absolutely right. Like JAK inhibitors and RUX has been around for a very long time, and probably something worthwhile digging in from the heme data. But
Yeah, it might be a quick chart review. It might help consolidate the rationale for the entire.
Absolutely.
Hey, Pavlos, congratulations on your first patient being dosed. I had a question about the TROP2 staining. Have you looked at quantitative continuous scoring like they did in the ADVANCE-R study for datopotamab to see if that's a better selector, because that's looking at TROP2 localization? And the second question is, do you just get a sense of whether RMC is more sensitive to topotecan inhibitors or topoisomerase inhibitors versus like MMAE payloads?
Great question. So this is something that we brainstormed quite a bit when we were designing this trial. How much should we make this be more biomarker enriched, given the fact that in the TROP2 ADC targeting space there is a lot of nuance that is being understood right now. To your point, we decided, at least for now, to allow all comers to enroll, given the fact that we showed in the Cell Reports Medicine paper that RMC tends to have generally robust TROP2 expression. But as the data are being gathered, we're ready to change that as we go. From our preclinical, granted this is in vitro data thus far, we believe, and there may be some clinical experience empirically, that topoisomerase 1 is a better payload for RMC. But of course, we're open to being refuted. That's how we learn. But the current hypothesis is that this one is better.
Darren.
Two questions. First for Tony. What was the rationale in thinking about why axitinib was chosen as the VEGF receptor TKI partner in your study? And then I have one for Dr. Litt.
Why do you think? I'm going to ask you.
I'm asking you.
It's very easy. Pfizer owns axitinib. It's an active drug. It's easy to combine. It has a short half-life. That's the simple. It's easy regulatory. Is it my favorite? It's not unreasonable at all. So it's a good question. But I think the deep dive, because Pfizer owns it. That's the answer that people generally don't give you, but I can give you. Ipilimumab, it's very reasonable. That's the only target you haven't had. So you have PD-1, VEGF, so what is left? CTLA-4 in general, outside HIF-2. I wouldn't be surprised if there is another cohort that comes up with a HIF-2 inhibitor, and that you have to pick the winner. I mean, they all have, in a way, single agent activity. I mean, it's a good and obvious question.
Since you're at the podium, Tony,
wait.
The vertical inhibition of VEGF and VEGF TKIs a long time ago, we had some problems with microangiopathy.
Yeah.
Absolutely. So a long time ago, actually, the Memorial Group with Bob at that time tried to combine sunitinib and bevacizumab. And also there was a combination, if you remember, of sunitinib and temsirolimus, but I would say we gave up early on that. Nobody wanted to do the IV weekly schedule, but sunitinib and bevacizumab, which is extremely, I would say, effective and high response rate, but it led to significant hypertension, significant HUS, TTPs, and all the complications. Now, in retrospect, could we have gone and selected the right population, optimized their blood pressure, etc., and saved that regimen? Too late to say. Now, are we gonna encounter something like this? At least we're using a drug that is effective, that have a short life. That's something to watch for sure. We're gonna watch the CBC. We're gonna see if there is any, smears. It's the thing when you talk to folks about this, if you have a toxicity from the bispecific that is VEGF related, where you have to stop the drug, let's say proteinuria, or an immune-related AE, you have to stop the drug, both of them. While in renal cell, in the combination of PD-1 and VEGF, whether Cabo-Nivo, Pembrolizumab, Pembro-Axi, you can stop one and continue providing some benefit to the patient. So this might or might not be a problem. We'll see.
Thanks for mentioning our study, but yeah, we did see a lot of microangiopathic hemolytic anemia actually in that in that study was sunitinib plus bevacizumab.
Oh, yeah,
we did. I was
you're
the first
author.
Correct.
You were a fellow.
Yeah. Yes, that's
I
right.
remember, yes.
But, and it was a lot of it was hypertension driven, but I think there were even cases we had that didn't, where
But
the
the
hypertension
response
was
rate
controlled. It was very high.
Very high, higher than what sunitinib give you, I mean, in theory alone.
Definitely high response rate. I have one more question for Dr. Litt, which is, that's a great study. This is what patients want, to come off treatment and consolidative surgery and really going for a home run. My only question is 6 months of immunotherapy enough in the adjuvant? You know, after resection, did you consider giving more adjuvant immunotherapy?
Thank you for the question. I think that's a really good question. You know, I don't think we know for certain if that time is adequate for every patient. I think there is some data that suggests that if patients do receive at least 6 months of therapy, they probably will, most patients will derive most of the benefit they were going to get for immunotherapy. We do know too that immunotherapy can sort of stick around for a long time after cessation. So certainly, you know, there may be some patients who do progress afterwards and won't go back on immunotherapy pretty quickly, but we at least wanted to see how long patients could last to be able to at least avoid some of the burden that comes with being on treatment, like the time and financial toxicity. But it'll be interesting to see what we find.
I think we gave a wiggle room because of nervousness, you know, up to a year. I just wanted to point out there's actually two consolidative approaches here. One is hitting everything before treatment, and the other one is doing the treatment and hitting it after the treatment with these two presentations. And I had one quick question for Rana, just that we use IL-6, you know, a lot of times a JAK inhibitor to address toxicity, but also to look at responses in cellular therapy. So what do you think about the timing and the usefulness, you know, PD-1 versus cellular therapy for using a JAK inhibitor?
You mean using a checkpoint inhibitor as opposed to using like a, you know,
no,
this is actually a combined JAK1/2. So Rux targets both 1 and 2.
Any other questions?
Yeah, so this is not a question, but I just wanted a shout out for Pablos. I feel that RMC is a very rare disease, and this community has really benefited from your efforts and also your keenness in clinical trial design. So keep it up.
So we'll stop here, and I think we're going to break for lunch.
Just one second. Well, first of all, this is the stuff that patients just eat up. I mean, we can't be more thankful for trials in progress, so appreciate that. Being in DC as much as I am, I got to make a comment. Aberrant nuclear speckle states. That's got to have a great acronym somewhere in there, but I like that phrase. But still have an exciting afternoon for everybody. Please come back for highlights of this year's major oncology events. Get outside if you have a moment. Pick up your posters if you've forgotten. Don't forget to take a photograph over at the photo booth, and especially if you're an institution, we'd really love it if you got all your folks together and went over and took a picture. We're going to begin promptly at 1:15, right back here, so go enjoy your lunch. Thank you.